Welcome to this video on the GCSE Biology required practical for food tests. We will cover the 4 key tests for identifying reducing sugars, starch, proteins, and lipids, including the necessary methods and important exam tips. Why do we test for food? By using specific chemical reagents, we can identify which nutrients are present in a sample. A positive result is indicated by a color change. Knowing these methods and potential negative results is essential for your exams. Here is an overview of the 4 tests. Benedict's test detects reducing sugars, the Iodine test is for starch, the Biuret test checks for proteins, and the ethanol emulsion test identifies lipids. The Benedict's test is used to detect reducing sugars, like glucose, maltose, and fructose. These sugars are identified by their ability to donate electrons or reduce other chemicals. For the Benedict's test method, prepare your sample, add the Benedict's reagent, and heat it in an 80°C water bath. Heating is essential for the reaction to occur. Never use a direct flame. The color change in the Benedict's test indicates the concentration of reducing sugar. A blue solution is a negative result. A green, yellow, orange, or brick red color indicates increasing amounts of sugar, with brick red showing the highest concentration. When answering exam questions on the Benedict's test, always state that the starting color is blue and that a positive result produces a brick red precipitate. Remember, it specifically tests for reducing sugars, not all sugars. Next is the Iodine test, which is used to detect starch. Starch is a polysaccharide, serving as a key energy storage molecule in plants. To perform the Iodine test, prepare your sample and add the iodine solution. Unlike the Benedict's test, no heating is required, and the result is immediate. A white tile can help you see the color change clearly. For the iodine test, a negative result remains orange-brown, while a positive result turns blue-black. This shift happens because iodine molecules get trapped in the starch's helical structure. Key exam points for the iodine test. It works at room temperature. It is specific only to starch, and precision in your color description is vital. Use orange-brown and blue-black to ensure you don't lose marks. The Biuret test is used to detect the presence of proteins. It works by reacting with the peptide bonds that link amino acids together in these large molecules. To perform the Biuret test, add the sample and then the reagent, which consists of sodium hydroxide and copper sulfate. No heating is required for this test. In the Biuret test, the solution remains blue if the result is negative. A positive result turns the solution lilac or purple, signifying that protein is present. For the Biuret test, remember the starting color is blue, and the positive result is lilac or purple. Ensure you do not confuse this with the Benedict's test, and note that heating is not required. Now let's discuss testing for lipids or fats and oils. The ethanol emulsion test is unique because, unlike the others, it relies on physical properties rather than a chemical color change. To perform this test, crush the sample, add ethanol, and then pour the mixture into water. Because lipids cannot dissolve in water, they form tiny droplets, creating a milky white emulsion. No heating is required. For results, a clear solution indicates a negative result, meaning no lipids are present. A cloudy white emulsion is a positive result. The cloudiness is caused by lipids, which are immiscible in water, dispersing as fine droplets. Remember these key exam points. The test is a 2-stage process. It relies on a physical rather than a chemical change, and it is carried out at room temperature without any heating. A common exam question involves knowing which tests require heating. Remember that only Benedict's test requires heating in a water bath at 80°C. All other tests, iodine, Biuret, and ethanol, are performed at room temperature. Here is a summary table for all 4 tests. Memorize the molecule detected, the reagent used, the color for positive results, and whether heating is necessary, as examiners often ask for this information. Valid and reliable experiments require you to control your variables. A fair test ensures that only one variable, the food sample, is changed, while others are kept constant. Key variables to control include the temperature of the water bath, the volume of reagent used, the concentration of your food solutions, and the duration of each test. Controls are essential. A positive control contains the molecule you are testing for to verify the test is working. A negative control, typically distilled water, confirms no contamination or reagent error. When recording results, use a clearly labeled table. Be precise with your color descriptions. For example, write blue-black instead of dark, and ensure you always record negative results too. Mastering exam strategy is key to securing full marks. We'll now look at how to approach common types of questions on these practicals. Examiners look for the correct naming of the test, clear descriptions of both starting and final colors, mention of heating where appropriate, and a correct, step-by-step method description. You will often be asked to complete a table, describe a full method, identify a molecule based on a color change, or explain the purpose of a control experiment. To finish, here is a quick-fire revision table for all 4 tests. Practice writing this from memory until you can complete it without hesitation. Precision here is worth multiple marks. The food test practical is a core component of your GCSE Biology assessment. By understanding these methods, variables, and exam strategies, you will be well prepared for any practical task. Good luck with your revision. To excel in your practical assessment, you must demonstrate 4 core skills. First, planning, identifying the correct tests and keeping variables controlled. Second, execution, following methods and reagent sequences accurately. Third, recording, using scientific language to describe color changes. And fourth, evaluation, commenting on reliability and suggesting improvements. Safety is paramount in any lab environment. Remember these key hazards. Ethanol is highly flammable, iodine stains, sodium hydroxide is corrosive, and hot water baths can cause burns. Always wear your lab coat and goggles, and never point test tubes at yourself or others. Unexpected results happen. A false positive might mean contamination, while a false negative could suggest low concentration or poor heating. Improve your experiment's reliability by repeating tests at least 3 times to calculate an average, which helps minimize the impact of any single anomalous result. Understanding the biological context is key. These molecules are not just test subjects. They are the building blocks of nutrition. Knowing the role of carbohydrates, proteins, and lipids in your body helps you answer complex, long-answer exam questions with confidence and depth. Think about their functions. Reducing sugars provide rapid energy for respiration, starch is a vital energy store in plants, proteins facilitate growth and repair, and lipids provide long-term storage, insulation, and protection for your organs. Use this checklist to track your progress. Can you name the reagents? Do you know all color changes by heart? Are you comfortable describing the full methodology and explaining the purpose of control experiments? If you can tick these off, you are exam ready. Here are 5 final exam tips. Benedict's is the only test requiring heat. Always state both the starting and ending colors. Use precise scientific language like brick red or cloudy white emulsion. Describe the 2-stage ethanol test clearly. And finally, memorize your summary table completely. Let's try a practice question. If a sample turns orange with Benedict's, you can conclude a reducing sugar is present, but at a moderate concentration. It isn't brick red because the sugar level isn't high enough. Remember, interpret the intensity of the result for full marks. For the iodine test on bread, remember the steps. Place on a white tile, add drops of iodine, and watch for the shift from orange-brown to blue-black. This confirms starch is present. Examiners expect this precise, step-by-step description to award you all 4 marks. You are ready for your exam. Remember, 4 tests for 4 molecules, colors are critical, heat only when necessary, and always control your variables. Review these concepts until they are second nature. You've got this.